Our next speaker is Tony Wei, and he is an assistant professor in the Department of Molecular Metabolism at Harvard University at the T.H. Chan School of Public Health. He got his PhD in biophysics from the University of California, San Diego, and then he went and did a postdoc in the laboratory of Joshua Rabinowitz. He joined the Harvard Chan School in 2020 and takes the approach of metabolomics and in vivo isotope tracing to study diseases of energy imbalance, including obesity and cachexia.
So our work is still in early stage, so I don't have a complete story to share today, but hopefully our approach and some of the initial observations can be of interest to you so that you are not wasting your time for the next half an hour. And for me, importantly, this is an opportunity to introduce myself to the bigger community of cancer research so that I can get feedback, also get engaged in discussions, which I'm looking forward to in this conference and also after this conference. So the work I'm going to talk about today is being done by two amazing postdocs in the lab, Gaoyang Huang and Yenshan Liang. And we benefit a lot from close interactions with the rest of the CANCAN team, including especially from labs in Project 1. David is part of Project 1. The focus of Project 1 is metabolic aspect of cancer cachexia. I'm very much grateful for the financial support from Cancer Research UK and the National Cancer Institute. So I'll start with the animal model. We use the colon carcinoma 26 or C26 mouse model. And we follow the protocol by Dr. Badato. We inject C26 cells subcutaneously to CD2F1 mice, which is the hybrid mice from BALP-C and the DBA2. As you can see in the red curves in these plots, this model has very robust phenotype, showing in body weight, fat mass, and lean mass. And for control, we also got from Dr. Bocciani another cell line. It's called CT26, which has the same origin as C26. And this cell line doesn't cause cachexia. It is showing the blue data points. So we use this as a control. This cell line grew a little bit faster in mice, so we inject half of number of cells at the C26 so that we have the similar tumor growth curve. So with this model, we want to test this general hypothesis that tumors cause metabolic phenotypes, metabolic perturbations to the host, which contribute to eventually cancer cachexia. And from tumor to cancer cachexia, this is a long, complicated process. There are many steps. And metabolic perturbations definitely doesn't sit on one point along this causal pathway. For example, it can occur at a very early stage. We know fast tumor growth can utilize a lot of nutrients, glucose, amino acids. They can cause perturbation to the host in terms of those nutrients homeostasis, which then can contribute to the later stage systematic metabolic imbalance. And of course, metabolic perturbation happen at any stage, the cachexia stage. Cachexia, it manifests as a metabolic syndrome. There is an imbalance of protein synthesis and the protein degradation, and the TG synthesis and lipolysis are out of balance too. And there are reports that the metabolic cycling flux is going up, contributing to the elevated energy expenditure in cancer cachexia. There are also reports that wet adipose tissue become browning adipose tissues that are contributing to the energy expenditure also. So our rationale is by identifying all these unique metabolic features to cancer cachexia, we can target them to alleviate the phenotype of cancer cachexia. So with this general rationale, our strategy is to identify metabolic alterations in both pre-cachetic and cachetic stage using metabolomics and flux quantification approaches. We use MESPEL for metabolomics. We use stable isotope tracing in fusion to quantify fluxes in cancer cachexia. And specifically for C26 model, at day 8, it's when the body weight loss starts happening. So before day 8, we refer it as pre-cachetic stage, and after day 8 is cachetic stage. So the rest of my talk organized along these two sections, pre-cachetic stage and the cachetic stage. Okay? In the pre-cat stage, we use the time course metabolomics to review the early change of metabolism in cancer cachexia. And in the cat stage, we first did careful measurements of energy balance equation variables, because food intake, for example, can be a huge confounding factor for metabolism changes. So we want to control that because we want to review tumor intrinsic impact on metabolism. So our measurement actually shows that food intake reduction accounts almost for all the body weight loss in this model. So we controlled for food intake and measured metabolomics and flux quantification to review the tumor intrinsic changes in metabolism. So first, pre-category stage. We first did the time course metabolomics for serum samples. We collect the blood samples every other day from day 0 to day 12, the whole disease development process. And then we run metabolomics on these serum samples. Here shows the result in heat map format. There are 3 groups, cacatic C26 and non-cacatic CT26. So you want to spend 5 seconds to memorize this, right? C26 for cachexia, C26 is not for non-cacatic control. Okay? And we also have a saline injected non-tumor bearing control group. As you can see here, there are a lot of changes in metabolism in the cacatic mice. But most of them, the changes happen at later stage. The axis here is time from day 0 to day 12. For example, glucose, acetylglycine, their change happened at day 8. So for the purpose here, which is identify the pre-cacatic metabolic change, this is not super interesting. We want to identify the metabolites that show pre-cacatic changes. Analysis of data show that there are 10 of them. 5 are amino acids, and 3 are metabolic intermediates of amino acids, and the rest 2 are TCA intermediates. So basically 2 categories, amino acids and TCA intermediates. To give you a sense of how they change, I show the 2 TCA intermediates. So at day 8, alpha-ketoglutarate and fumarate, the level dropped by half at day 8, even though here there's not much change in body weight. So these are important candidates for contributing to later stage of systematic metabolic dysregulation, because this happened very early in the disease development. So next step for this would be to manipulate the level of these important candidates. For example, we can supplement alpha-KG to the diet to try to rescue the level of alpha-KG to see its effect on cancer cachexia. Another lab in our project actually saw the same job in their model, which is the GEM model. So they are doing dietary supplementation of alpha-KG to test its effect on cancer cachexia development. So my mouth tends to be very dry and then become a stutter. I need to drink. Okay, so that's the time course metabolomics for serum. What about tissues? Is there any tissue that show very early change in the disease development? So for that, we collect 14 different tissues from day 4 to day 12, again, every other day. Right? We run metabolomics for all these samples. The results will be something like this. And a lot of analysis can be done for this huge number of data set. But I'm going to just show you a very simple analysis result, which is the number of significant change in metabolites for each tissue or tumor or serum. So each bar, the length of each bar is the number of metabolites we can detect for that sample, tissue, serum, or tumor. And the length of this yellow bar indicates the number of significant change in the metabolites and end stage. So again, it's not super interesting here for the question because we want to identify the early changes. So which is shown in the red bar. So here, before day 8, not many things going on. For many tissues, there's like 1 or 0 metabolites that changed. For example, gastrocnemius, soleus, there's nothing, right? Nothing changed before day 8. But I guess you already noticed one tissue that stands out in this development of cancer cachexia is spleen. Before day 8, before anything happens, 40 metabolites change the level significantly in spleen. So this leads to an important hypothesis that spleen may sit at an early time point of the causal pathway of cancer, Cachexia. What are those metabolizing spleen? Listed here, and we did pathway rearrangement analysis for these 40 metabolites. And the top two are pyrimidine and purine metabolism. So nucleotide metabolism. So this suggests that a lot of DNA synthesis is going on, meaning a lot of cell proliferation is going on. So a simple hypothesis would be at the pre-cachexia stage, spleen is doing a lot of immune response. Okay, and anyway, so to follow up this study, the first experiment we want to do is to remove spleen from the mouse, see what happens after we inject cells. So that's basically the first part. I showed you that we identify a small number of metabolites that show early changes. They fall into two categories, amino acids and T-cell intermediates. They are very important candidates to follow up to test whether they have causal role in this cancer calcium development. I also show you that spleen has very early changes. It may play a causal role in cancer calcium development in this model. Next, I'm going to move to the cachexia stage. We first did energy balance measurements because food intake is a huge confounding factor for metabolic changes. If you don't control food intake, you see a lot of changes. It doesn't mean those changes come from tumor, right? It's just because they eat less. It's not very interesting. So we did that. So this is well known in this model that anorexia phenotype is very strong. And we measure food intake. It drops very, very strongly. And we first, to figure out how much food intake accounts for the body weight loss, we did a pair feeding experiment first, which is shown in this yellowish color. And it doesn't make sense, right? Because the body weight loss is more in the pair-fed group. You know, the non-cachexia group lost more body weight. It doesn't make sense. We conclude that this is an artifact from the pair feeding experiment because in pair feeding, you give the mouse in the evening a limited amount of food, right? All they see is a very small amount of food. Maybe they tend to eat very fast. And for the cachexia C26 mice, they see unlimited food in their cages. They may have different feeding behavior. So to control for that, we developed what do we think is a better pair feeding strategy. We call it isocaloric restriction. So basically, follow the food intake measurement. We gave both group, the C26 group and CT26 group, the same amount of food as every evening at 7 p.m. to be exact. And here, the result makes much more sense. You know, the cachexia group lost more body weight, but not much more. The difference is very small. This shows that food intake reduction accounts for almost all the body weight loss in this model. Just a very interesting side note is that when you limit the food for the CT26 tumor-bearing mice, the tumor gets smaller. But this is not super interesting for us as a cancer researcher. So coming back to the body weight loss curve, this indicates there's not much contribution to body weight loss from energy expenditure. So we directly test that by metabolic kg measurement. As you see here, for the ad-lib CT26, there's no change of energy expenditure over time. You know, they eat normally. But for the cachexia C26 group, their energy expenditure goes down. But when you look at the controlled group with food restriction group, they drop similarly. So there's no difference between C26 and CT26 when food is controlled. Okay, so this is consistent with the previous the pair feeding data, and the food intake accounts for almost all the body weight loss. Another important variable in energy balance equation is energy excretion in the form of fecal energy. So we measure that. Again, there's no difference between the C26 and CT26 when food intake is controlled. So if you don't control the food intake, you see a big difference. But if you control food intake, there's no difference in energy excretion. So to summarize, again, in this model, the body weight change mainly comes from a food intake change. So next, in our metabolomics and the flux studies, we control the food intake so that we really try to identify the tumor intrinsic or tumor direct effect on metabolism. First, I show you the serum metabolomics. The PC results show that it's important to do the right control because with and without food intake control, the metabolic profile is very different. Okay, this is it doesn't show up my. It's supposed to be a Venn diagram. So there's a circle here, another circle here. Okay. Okay. So. Yeah, so if you do the traditional comparison, which is the C26 compared to the CT26 at the lip without food control, you're going to have 41 significantly changed metabolites. But if you do the proper control, and you only have 25, the overlap is 20. Okay, so the 21, which is which is not supposed to be there, and the list includes, for example, keto bodies. Just to show you an example, the 3-hydroxybutyrate. If you compare to the ad lib, there's a significant increase of the 3-hydroxybutyrate. It doesn't mean in calcium that there's the increase of keto body generation just because they use less, right? If you compare to the control with food intake, there's no difference. So instead, we should really focus on these 25 metabolites. They reflect the tumor direct effect on metabolism. So, as I said, we're still in the early stage of our work. The next step would be, look at this list and combine this with other data like transcriptomics and also results from other models to really come up with specific hypotheses to test individual effect of these metabolites. But here, I just want to quickly point out some interesting observations. There are 4 TC intermediates here, so their level are dropped in CACAT stage with food intake controlled. And quinurenic acid and indolactic acid, they are both metabolic intermediates from tryptophan degradation, but they sit on different degradation pathways of tryptophan. The fact that one level is lower, the other level is higher, indicates there's a shift of tryptophan degradation pathways in cancer cachexia. So that's the result for the serum metabolomics and the CACAT stage. We also did the same thing for all the 14 tissues. Right here, for the sake of time, I'm not going to through all the tissues. I'm just going to show you our result for gastrocnemius. Again, the PCA results show that it's important to do the right control because the metabolic profiling can be very different with or without food intake control. Again, here it's supposed to be a Venn diagram, and 22 is not supposed to be there because this really comes from food intake. Okay? And we should really focus on these 11 metabolites. And these are very interesting because a lot of them are nucleotide metabolism and ribose metabolism, and the 2 of them are urocycle. So this is a very interesting features happening in gastrocnemius in CACAT stage. They're very important observations to follow up in the next step. So with that, I'm going to move to the last part of my talk, which is use SW tracing to identify flux alterations in CACAT stage. Okay? So first, we did the glucose infusion, and we calculated the glucose turnover flux. There's a drop of glucose level in the serum, but the flux measurement shows that there is no drop of flux, the turnover flux of glucose in the CACAT stage. The trend is even that there is a slight increase of glucose turnover flux. The glucose turnover flux is equal to the production flux of glucose and also equal to the total consumption flux of glucose. So if we focus on the consumption side, so we have a situation where the pool of glucose is lower, but the consumption flux is not lower or maybe even a little bit higher, meaning that there is a more efficient glucose utilizer in the system, which is probably tumor, because that's the difference between the cacatic and non-cacatic, obvious difference between the cacatic and non-cacatic mice. So this hypothesis is kind of tested positively in the labeling data in tumor. As you can see here, the labeling of TC intermediates in the C26 model, C26 tumor is much higher than in the CT26 TC intermediates, indicating that the Cachexia tumor burned more glucose. So to really directly test this hypothesis, we need to measure. The next step would be to measure the glucose uptake rate by the individual tissues and also tumors. This would be excellent application for Davis technology. So we also did a valine infusion because valine is the essential amino acids in fasted mice. The only source for valine would be from proteins, right? So the valine turnover flux will reflect the protein degradation flux. So here, interestingly, we don't see any changes in the protein degradation flux, which is kind of inconsistent with the gene marker measurement, right? The estrogen-1, the MORPH1. So I don't know. Next step would be to directly measure the protein degradation flux in individual muscle types, and also protein synthesis flux in those muscles to find out what's really going on. Okay, so I'm going to summarize the second part of my talk. I told you that anorexia accounts for almost all the body weight loss in CT6 model, and the controlling food intake reveals important candidates which reflects the tumor direct metabolic effects. They would be important targets for the next step of research. And the flux analysis shows that tumor glucose consumption may account for the reduced blood glucose level in the Cachexia mice, and there is no significant increase of protein breakdown in Cachexia. And just to recap what I told you early in the pre-Cachexia stage, we found a short list of metabolites in circulation, which may have a causal role in cancer Cachexia development. We also found that spleen has huge change before Cachexia happens, and it may have a causal role again in the cancer Cachexia development. I'm going to stop here and take questions.
That was a great talk. Quick question. I was really intrigued in your isocaloric studies about the changes in the tryptophan-based metabolites. Do you see any overall effect on NAD levels, since it's important for NAD, de novo production?
Yeah, good question. So yeah, there's so much data, and I put these slides together very recently, so I would need to look back. I vaguely remember I saw something, yeah, AND, maybe NDH. Yeah, I don't know. Sorry.
Thanks for a really great talk. In many of our hands, we see really significant expansions in the size of the spleen in the C26 model. In my hands, it's about 6 times bigger compared to a control mouse. Do you see that in your CT26 model, and do you see differences in spleen size when you do the isocaloric diet?
Yeah, I'm supposed to see that in my talk, so I did say that. Yes, but 6 times, probably it's more like 3-4 times for us. Yeah, pretty big increase, yeah. But I don't know at the day 8 pre-Cachexia stage how the size was. We need to do the measurement. But I suspect it's also a little bit bigger. Yeah.
It looked like you were only looking at polar metabolites. Did you examine lipid species at all in your analyses?
Yes. Yeah, we have run all of that. Yeah. So I didn't present any of this. Yes.
Hi, very nice talk. You mentioned no increase in energy expenditure. I could not see in your data a control group either on chow diet or isocaloric. Can you comment on this?
Sorry, I'm not sure I get a question. You said there's no increase of energy expenditure?
There's no increase in energy expenditure, but you were comparing isocaloric C26 and CT26. I could not see the healthy control there.
Yeah, both dropped. Both dropped. Yeah, but to the same level. Yeah, there's no energy expenditure increase in this model. Yeah,
you don't know the control reference, so the health control.